award winner. Our next speaker is Wellington Muchero, and Wellington has not been here quite as long as Melanie has. So Wellington actually got his bachelor's degree in 2003, and that was in California. And basically his entire career before he came here was in California. So we've moved him from the left coast to the right coast. So he got his PhD at the University of California, Riverside in 2007 and did a postdoc there, I believe, before he came to Oak Ridge in October of 2010. Wellington's an expert in applying genome-wide association studies, and that has led to a large number of publications with the plant sciences group here at Oak Ridge, including one in Nature Genetics. He's a critically important contributor to both BESC and PMI, our two largest projects in the biosciences division. He's also a contributor to several other projects, including US DOE, USDA joint funded grants. And what's unusual for people in BSD, he has 23 or more invention disclosures, mainly on discovery of the functions of certain genes in plants. Those have led to several patents. The patents have led to several licenses, and he's been funded by the TIP program, which is a technology program at Oak Ridge, to move our discoveries into the commercial realm. And most recently, he's been awarded the Early Career Award, and that's going to be the subject of his talk today. So thank you, Wellington. And
Tony. Thank you for the introduction, and thank you everyone for showing up today to celebrate with myself and Melanie after we've received this Early Career Award. It's really interesting that I spent the rest of my morning actually, let me see if I can regroup here. Instead of rehearsing my slides this morning, I actually spent more than a few hours wondering why life could be so cruel. It's like, why me? Why now? What did I do to deserve this? It's sort of, and I'm not talking about the early career and the 2.5 million. That's really fantastic. That part I'm happy about. Jay Chen and myself spent 2 weeks, the last 2 weeks, setting up plantations in the deserts of Arizona and Oregon. And it was a brutal trip. And just before we were about to return, I get this thing that shows up on my lip. It's sort of like you're getting ready for your first date and you get a zit on your nose. And then DOE insists that this has to be recorded for public viewing. So instead of rehearsing my slides, I spent my morning messing around with my wife's makeup, trying to see if I can, she doesn't know yet, so pray for me when I go back home that all will be well. But I think the moral of the story is you don't need good looks or flawless appearances to get an Early Career Award. And that part I'm really happy about, and I sincerely thank the DOE Office of Science, specifically the BER, for putting the voice of confidence in my proposal and suggesting that it is a worthwhile idea to pursue. And my only hope is that from now on, then I will be able to demonstrate and justify that confidence that DOE has shown. And I will echo what Melanie said when she started regarding when she came to ORNL, as a baby. And in a sense, I echo the exact same sentiment. I was a baby, not in terms of my age, but in terms of my scientific knowledge. I am a plant pathologist by training. I was educated in agronomic systems where you only look at one plant and one microbe at a time. Concepts of ecosystem level studies were non-existent to me and were virtually worthless. So, and over the past 5 years, I've had the pleasure of working with really gracious and patient colleagues and mentors who have driven me from that sort of really narrow-minded focus of research to where I can propose an idea at this scale and it's actually received with confidence. So as I walk through, it's not lost on me that my early career award was a result of collaborations and not my own thinking. And so the way I set up my presentation was to illustrate exactly that, what pieces of the EESD of our directorate that played into shaping my thoughts and actually providing valuable background information and preliminary data that I was able to present to be able to get to be at this point. So my work falls in the framework of DOEB, where we need to start understanding how genomes and the genes they contain shape protein, molecular interactions, cellular function, and how they affect assembly of communities, and specifically communities of plants and microbes. And we have 3 different main projects in ESD that fall nicely within those scales of biological study, and that's where the most benefit was for me to gain insight and to be able to make to come up with this proposal. And so I'll go through each and every one and highlight the components that played into my early career proposal. And the first one is the plant microbe interfaces that Jerry and Mitch lead, and I was thrown into as soon as I came to ORNL. And part of that was I was forced to interact with microbial people from the microbial world, where I'm a plant-centric person, but I had to listen to their gripes and complaints about how plants don't do anything but just sit there. It's the microbes that do everything. I sat patiently and listened to what they had to say, and I respected it and actually learned a lot. And so one thing that came out of the PMI, or that continues to come out of the PMI project, is the understanding that plant microbe interactions are not random. These are not random events. They are actually driven by highly specific molecular processes that are derived from both the host and the microbe. So it's a complex interaction that's going on that actually mediates this interaction. And PMI has opened eyes at this level where we are characterizing what those molecular mechanisms are on the host side and on the microbe side. And on the applied side, this is not only just fundamental research. We have actually demonstrated in PMI that you can identify plant growth promoting microbes. And Jesse Labe, my colleague, Sid Hamilton, and Udaya, they are demonstrating up to 180% increase in biomass yield just by visually introducing couple microbes in the system. So there's actually really strong application beyond understanding the fundamental mechanisms. There is applied knowledge that we are gaining from the plant microbe interactions. And so that was really one of the key sort of where I started with this proposal, because the core was specifically for plant microbe interaction and how they affect bioenergy production. And so that takes me to the next research entity that we have here that Paul leads, and that I have been really fortunate to be a part of. And this is how, looking at carbon dioxide as a greenhouse gas, we always look at it, from the negative perspective. It's going to cause global warming, and we are all going to die, and that's not good. But within BESC, we recognize that actually carbon dioxide can be a valuable commodity. Plants fix carbon dioxide, and they make biomass, which is what you see here from this schematic. That biomass is made of valuable polymers and valuable monomers that we can then deconstruct and make renewable energy or renewable bioproducts. And so if you think about it, if plant growth promoting microbes can increase biomass yield, we can increase the efficiency of biofuels production by simply increasing how much biomass you produce per unit area. So that's a really one way of linking BESC and PMI. And at the same time, while you are using these growth-promoting microbes, you don't need to use synthetic fertilizers that are toxic to the environment and energy expensive to manufacture. Now we are offsetting the use of synthetic fertilizers with the use of plant growth promoting microbes. So now we have two different benefits that we have from understanding the interaction between plants and their associated microbes, and then moving into and expanding into our renewable industries. The third component, which has always been sort of like on my radar and a really curious part, and this comes mostly in my discussions with my colleague Dave Weston, who has historically been the link between our group and the climate folks. And within the context of the NG, the next generation ecosystem experiments, there's always this talk about how they observe, especially in the Arctic region, as a result of the warming and the receding ice. You see these plant communities that are marching forward, following the ice as it moves back. And so those processes, that community expansion of plants has to have some impact on carbon cycling in that vulnerable region. And Stan and Rich Nobby had extensive discussions, and I've benefited a lot from listening to them talk about these processes, where the role of plant and microbe interaction in carbon cycling has been recognized as key. But the next step that has been not fully explored is how can we link in genomic studies in plant and microbial communities. How can we link that information to the ecosystem processes that NG specializes in? So I recognize that as a really nice, unique tie-in to the work that we've already been doing within the BESC organization and within the PMI project. So part of that then was, we recognize this opportunity. We have this tremendous potential to harness plant microbe interaction for the benefit of society, both in terms of economics, fuels, bioproducts, and also environmentally mitigating greenhouse gas emissions and global warming. But there's always a caveat with ecosystems and biology. It's always a mess. And as I presented before, I said plant microbe interactions. That's assuming that they always interact. In fact, most of the times they don't even interact. So that presents one of the biggest barriers, where plants, because these are perennial plants that were designed by and shaped by their environment to survive for decades, sitting in one spot. They cannot run, they cannot hide, they cannot take medication when, they cannot beat off a bug that's chewing on them. So they have to be pre-programmed in their genomes to tolerate whatever comes at them. And part of that is a really robust immune system that soils are in direct contact with dirt all the time for 100 years. And yet the plant remains healthy. It's a result of the innate immunity system that the plant has evolved. And we have illustrated the impact of that innate immunity with our work collaborating with Chris Schott and Melissa Krager and Dale Pelletier within our PMI project. And here I'm showing the interface that I'm talking about. So this is your dirt, your rhizosphere. This is your cell wall, and this is the inside of the plant. So you see you have this many microbes that are sitting right next to the plant, but only this few can make it within that barrier. And that barrier is not a solid block of steel. We know it's porous. Water moves in, nutrients moves in, exudates move out, signals are secreted. So it's open. Why can't microbes penetrate that? This is the defense that we're really focusing on. And in PMI, we have made some really interesting discoveries that have highlighted sort of like what is the mechanism of that, and how do plants and microbes modulate that defense to be able to establish a functional presence inside plants. So I show one specific example where we successfully showed this is a PCR assay showing each lane represents hybrid progeny from a cross in Populus deltoides. And what you see is they segregate for the presence of that DNA band. And that DNA band represents a whole gene, a membrane-bound receptor that segregates in that protein. Where you don't have it, it's a complete deletion, so you don't have that gene. Where you have a band, you have a functional copy of that gene. The consequence of that is whenever you have the gene, you have twofold increase in interaction between that host and the microbe. Where you don't have it, you really have poor colonization. So from a plant pathology perspective, this is counterintuitive. You think genes are there to protect the plant, but actually these genes are there to suppress the defense response so the microbe can interact with the plant successfully. And we thought this was really exciting. And we took it with Jay Chen, who does the transgenic work in our group, expressed it in Arabidopsis. It goes where we know it should go, which is on the membrane, where it can interface with the environment. And we see changes in interaction with the microbe, and we can actually see evidence of fungal penetration within the intracellular tissues of the plant. And this is only by introducing one gene. And to actually demonstrate this defense suppression that I'm talking about, here the same plant, we took it from Populus and we put it in Arabidopsis. So I'm showing the Arabidopsis wild type without the gene and without the microbe. And this is the Arabidopsis wild type without the gene plus the microbe. And you see huge expression of this pathogenesis-related one gene, and it's a marker gene for defense responses. It's sort of like when you go to your doctor, they have to have some way of evaluating if your body is responding to some infection, right? Usually white blood cells or something like that. So this is sort of the equivalent of white blood cells in plants, where the plant recognizes it's been attacked, initiates a defense cascade, kills off the microbe. This is what happens when we put in that gene in the same plant. You actually mute the entire response for pathogenesis. So the plant responds like nothing is happening. And the microbe just goes gangbusters, takes over, does whatever it does. And in this case, it's actually good for the plant because then it helps the plant grow. So this is where PMI has taken us, the cumulative knowledge that we have gained so far. And my interest was, how do I move on from what we have discovered now and actually come up with a project that's relevant for biofuels and bioproducts and also relevant for climate change mitigation. And a really nice species came to mind, which is Salix, a close relative of Populus. It is widely used biofuels feedstock all over the world. It's used for environmental remediation. So it is a perfect avenue for discovery and deployment because it already has utility in our community and in our societies. But not only that, in the Arctic region where the shrub expansion phenomena is occurring, it is the only shrub that has a reference genome sequenced. And this is thanks to work that actually was the work that originated here at ORNL with Jerry Tuscan and Jeremy Smarts at JGI, and Larry Smart, who's going to be a collaborator with me, and Steve DeFazio, where we have 2 reference genomes for this species. So it's a perfect avenue to start putting some of our discoveries in there. And why it's really important now is this is a single genus that's widely distributed, and it exhibits wide variation. You can see in terms of biomass accumulation, this is at Cornell University where it's grown for biofuels. And this is actually an image from the NG research project where they find this Salix rotundifolia in their experimental sites. So we can apply our knowledge, or at least our research, in both directions for bioenergy and also for shrub expansion. And my hypotheses are actually centered on this, the genomic adaptation to different environments, why a single genus is highly successful across very different environments and across very different applications. So to me, I thought Salix would make a perfect model. From now on, it's only going to be text, and I apologize for that, but this is sort of the mind dump that I had reviewers deal with. My specific hypotheses are these membrane-bound receptors that suppress host immunity have to recognize specific microbial features because they are so specific in how they interact. They don't just recognize any microbe. They are very specific. Otherwise, they will be allowing pathogens alongside with beneficial microbes. So there has to be a very specific discrimination that's going on there. And I'm interested in finding out what that would be. And as Salix moves from different environment to different environment, it's going to face new, novel partners, interactors, threats. And so these receptors have to evolve rapidly. And we already know that in genomes, they typically occur as tandem repeats, and they show high levels of duplication and deletion as speciation occurs. And so as this speciation has occurred with niche separation, then we will be able to see highly species-specific interactions. And if we can, heterologous expression of these can be used to engineer non-host into new host, as I illustrated with the case with Arabidopsis. In this case, when we're actually looking at same species, you can pretty much just hybridize. This is sort of like classic breeding that you can do. You can either use breeding approaches or transgenic approaches to incorporate those. And so the approaches that we'll take are simply, the beauty of long-read sequencing has enabled us to characterize those tandem repeats and large structural variation much better than we used to be able to do with Illumina-type sequencing approaches. And then with the, sort of like I mentioned before, that I set through gripes from the microbial folks, I will actually still lean on them to characterize the different microbial communities across these different environments, different species, and see if we can now start creating interspecific crosses and then generate the exact same pattern of PCR products that I showed you, where you have presence, absence of these receptor kinases. And after you do that, can you link the presence, absence of the receptor kinase with presence or absence of specific microbes? So that's where this will take us. And then our final approach was to perform transgenic validation, where we take that gene and put it in a non-host and see if we can recapitulate that interaction. And then opportunities for fundamental discovery, and this is where I'm really excited to be part of ORNL, and we have really amazing core capabilities here that will play really nicely into finding out what are these microbial features that are recognized by these plant genes. And what is it about this? If you look at the protein structure of these receptor kinases, they are highly identical, and yet minor variation completely changes their specificity. So what is the characteristic at the protein level that confers that ligand specificity and that discrimination ability that I talked about earlier? And from then on, can we build models where we can predict from DNA sequence alone or protein sequences and say this is the structure that the receptor kinase is going to form, and it's going to recognize this microbial moiety to be able to, to facilitate interaction. And from that point on, we will be ready to then go back into the complex ecosystem and start building on, instead of doing single pairwise comparison, multi-level comparisons between host receptors and microbial features to see if now we can still, like the illustration I showed from DOE, working at the community level instead of the individual genome level. So that's where I hope this is going to take us, and I'll be very excited to interact with as many people as possible. And lastly, as I mentioned earlier, my experience here has been shaped by many projects, and I have the joy of interacting with many other institutions outside ORNL, and that all has contributed towards the success of the proposal that I put in. And I thank ORNL, the leadership team, specifically Jerry Tassin, my immediate supervisor, for his constant thoughts and wild ideas and crazy, well, we spent the last 2 weeks in wandering the desert for a reason. It was because of him, but good things do come out of it. So thank you very much for your attention, and I'll take any questions.
Any questions for Wellington?
Congratulations. It's very cool.
Thank you.
So I spent a lot of my career trying to run away from microbial people too, and I failed also. So maybe we can share stories on how we might have done that differently.
Yeah.
Or maybe we should just thank them because they're everywhere, whether we like it or not.
I know.
Both.
Both.
So again, non-technical question. As you kind of look at how you came here with the plant focus and you've learned to interact in these different ways, what was the hardest part about learning to work across collaboratively with folks in the different disciplines, and how did you overcome that?
I think for me, the first time, it was intimidation. I mean, they sound really confident about their stuff. And they will dismiss your field easily. But well, first of all, I think, one, it has to be a personality. you have to have and Melanie said the same thing, you really have to be open to interacting with people. And I know they've done the same thing for me. When I interact with them, they feel the exact same way. So it's sort of like a two-way exchange between personalities and needs. And when I finally realized that I just had to survive this was when I recognized the value of what they were doing and how it would inform my own science. Because I could never have we could never have discovered these receptor kinases if there was no microbial community characterization. So it's just realizing that you cannot avoid that expertise you needed for your own science to succeed. And then at the same time, I think you also have to bring value to what they are doing, where you bring exciting questions that they can also explore, such that we are pursuing research side by side, but one is just not pushing the other forward. But it's a mutually beneficial interaction. And I think I've had that specific interaction both within PMI, where we interact with the microbial folks, and then within BESC, where we interact with folks from chemical engineering, who have no clue about biology. So one, openness, and two, recognizing value in what the other group brings, and three, recognizing what your value is to them as well, so you can show them that they need you too.
Wellington, from an evolutionary point of view, you talked about how perennial plants, herbaceous and woody materials, just stand there and have to take it, over decades or over a century of time or longer. From an evolutionary point of view, how do you think plant-microbe interactions have impacted the way genomes evolve in perennials versus annuals? The idea of de novo gene occurrence,
right.
The co-option of motifs, and do you think that's going to be a story that will have a universality to its applications, or is it something specific to the systems you've worked in so far?
That's actually a really good question. And the one main distinction that we already see is when we look at annuals versus perennials. So in annuals, you don't see this huge, rapid duplication of these genes in tandem repeats. They typically have far fewer copies of receptor kinases than perennials. And the reason is because they only have to survive for a season. And they have safety when they form seed, and they germinate again, survive for a season. So their exposure is not as constant as the perennials. So the perennials then, the genomes are driven by microbes to rapidly evolve in these tandem repeats for specific genes. And also, as you mentioned, we see evidence of de novo gene evolution where non-coding sequence evolve within a species to new functional genes. And those functional genes keep showing up each and every time we do RNA-seq analysis where we overexpress one of these receptor kinases. You see those, typically they are called orphan genes because you don't find them in any other organism, but the only one they evolved in. And they are highly related to disease resistance and microbial community interactions. So one of the key points of this entire project is going to evaluate that rate of genome evolution across different species and across different environments as they are exposed to different microbial and environmental factors. So yeah, that's one of my, hopefully we can make progress on that line of reasoning.
Very, very nice job, Wellington, and congratulations again. Can you speak more to the relevance of your project to the climate change side of the question? Obviously, biofuels have a direct relevance to the management of carbon for mitigating climate change. Right. But in terms of what we might learn about plants in regard to their response to climate change and that sort of thing, could you speak more about that?
Yes. So part of the setup that we have here is working closely within the NG research sites. So in addition to coming up with really specific objectives of what we want to achieve, one of my thoughts has always been, and I think I spoke to that earlier, is as has been observed, as this ice is receding, the plant communities are following, and specifically shrubs, woody shrubs. So the benefit with woody shrubs is longer residence time for carbon dioxide when it's fixed. Beyond that, I don't think we really understand, as Jerry's question actually directly plays into that, what is changing within the genomes of these shrubs as they are moving from their old colonies to their new world. Are they changing at all, or are they just dragging microbes they were interacting with them historically and bringing them to the new environment? Are they forming new associations? Are there some novel microbes that were already under that ice that all of a sudden are rejuvenated and now have opportunity to interact with the plant communities? So those dynamics I don't think have been looked at or are understood. So from the plant side, I want to know what's happening to the genomes at the leading edge of the shrub expansion versus the rear end of it. And from the microbial side, are we finding new microbial associates that were not found at the rear end but are actually present at the leading edge of that shrub expansion? That's one immediate thought that I have in my mind that I think we can go after fairly quickly within the context of the 5 years.
This is really interesting, and I'm so glad you get to continue this work. I was really interested in your comments in response to Jerry's questions. You were talking about annuals versus perennials.
Right.
And I wondered what the implications of that kind of evolutionary thinking have to do with disturbed areas versus not disturbed areas. For instance, in some sense, a lot of the places where humans are is highly disturbed, and sometimes the soils are greatly disturbed.
Right.
And so when you have plants coming into that, should we manage them in different ways as compared to places where we're maintaining systems in a way that they have been? And this goes with climate change because we may be changing disturbance or even other more dramatic disturbances. So do you have any thoughts about microbes and plants and disturbance patterns?
Yeah, so I think one of the interesting, I remember a specific example about disturbances, and it had to do with mining and soil contamination, and this was with a plant, Mimulus. I'm not very familiar with its biology, but they found actually that by mining practices where they were dumping toxins in the ground actually drove rapid evolution of metal transporters in those Mimulus that were found in those specific areas. And when you looked at the neighboring communities that are actually not distant at all, they didn't have that rapid expansion of metal transporters. So I think it does that we need to have a clear understanding of how man-made disturbances affect plant genomes and performance after that. So are we driving evolution of specific genomic features that are beneficial or may end up being detrimental? Or are we prejudicing sort of like this species to its terminal point by not taking into account the specific considerations that you just mentioned? So indeed, I think the disturbances that we make on soils and on communities have a really strong implication on the plant and its performance, and maybe its eventual participation in remediation of these environments. So I don't know if that gets to your right.
Yeah, the evolution questions I think are really interesting. I'm curious, you talk about the plant microbe interactions, which are largely a root soil interaction
Right.
environment. And the de novo genes, expressions, and all those interactions, do those find their way into the germplasm of these perennial plants, or is it strictly somatic? And if it's somatic, does it find its way into the vegetative reproduction?
Interesting question, actually. And I'll let Jerry also dive in at this point. We actually had a project where we looked at that specific question about somatic mutations. And within the same tree, which is about 300-year-old trees, you go by different branches, and you can age the branches over decades. And you do find somatic mutations that occur. And so we suppose, so somatic mutations do occur in these tissues. But for the orphan genes to be successful, the mutations have to occur in germinal tissue. And they have demonstrated that with plants and with Drosophila, that the only way you can get a true orphan gene that's actually then find its way into the germplasm if it occurs within reproductive tissue. And there's actually evidence that the most orphan gene formation happens in those type of tissues. So it looks like there's an evolutionary consideration already that if you are making these things, you need to make sure that they go to the next generation instead of die, with the parent.